Hello everyone, my name is Yannik Roell, and today I'll be talking to you about an empirical test of the role of topographic complexity in the general dynamic model of oceanic island biogeography. This work is part of my master's research at the University of Idaho. Before I get to the general dynamic model, I'm going to quickly mention the theory of island biogeography by MacArthur and Wilson. So what this is looking at is we're looking at species richness on islands. So the two things that they're using to predict species richness is isolation of the island from the mainland and the area of the island. So islands that are more isolated are going to have fewer species immigrating to the island, and islands that have less area are going to have a higher extinction rate. So with these two together, you can see where they overlap, and that's the predicted species richness. So islands that are small and isolated are going to have fewer species than islands that are closer to the mainland and larger. So I mention this because many models have been built off of this, and one of them is the general dynamic model of oceanic island biogeography, or from now on I'm going to refer to it as GDM, by Whittaker et al. And what they use is area and time. So many studies have seen that while isolation is important for islands, it's not isolation from the mainland, it's mainly isolation within the archipelago. So they use area. There's a linear relationship. As area increases, species richness is going to increase. But for time, they have a hump-shaped relationship. And the reason why they make this assumption is because volcanic islands follow a very typical lifespan. So an island will emerge from the water, have a quick period of rapid growth, where the island will reach its maximum altitudinal range and area. And then after this volcanic activity has ceased, the island will slowly erode until it loses all of its area and altitudinal range and submerge back under the water. They make an assumption that topographic complexity will follow a hump-shaped curve, and its maximum will be shortly after the quick building period. And while in the model and a lot of other studies have kind of verbally agreed on this, there's not any study that has quantified what topographic complexity is. So there's many different ways that we can look at how a landscape is complex. So in this study, what I'm looking at is, does topographic complexity follow a hump-shaped curve? And if we use a complexity index in the GDM, does it help predict species richness? So all of this work is being done in the Galapagos. So this is about 1,000 kilometers off the coast of Ecuador. So when we zoom in, we see that there is a progression of age with the islands, where in the southeast islands, they're the oldest, and the west islands are the youngest. And this is important because we need to get three things first. We need to get age, isolation, and area. So age, we have the age for each island from previous studies, and then using a GIS system, I use ISRI's ArcMap. I used a pairwise distance for the isolation. So I take the one island and average distance to all other islands, and that's the isolation metric. And then also within a GIS system, you can get area pretty easily. So once I got that, I got the species and complexity indices. So for the species, I got all my data from the Charles Darwin Charles Darwin Foundation data zone, and I downloaded all the data for terrestrial and native species, for plants, vertebrates, and invertebrates, and then broke vertebrates down further into birds, mammals, and reptiles. And then another measure that many people look at species richness is looking at single island endemics, so where a species is only restricted to one island. And I looked at plants, vertebrates, and invertebrates. There's another data set in the lab that I'm in, Nasiotis, which is the group of endemic land snails. And I incorporated this because while the Charles Darwin data zone only has data for 14 islands, we have data for 25 islands. And then all the topographic complexities are based on elevation, slope, curvature, So for this presentation, I'm going to show the main results for the snails and then kind of sum everything else up. So to get the complexity indices, sweet, okay, perfect. Okay, so to get the complexity indices in ArcMap, we created 4 maps, one for elevation, slope, curvature, and viscosity. So 4 really common complexity indices used are range and standard deviation of elevation, and range and standard deviation of slope. So what you do is, let's say for range of elevation, you take one island here, you take the maximum of elevation, the minimum elevation, get the range, and let's say that is 30 meters. So if an island has an elevation range of 100 meters, you'll say that island is more topographically complex than one with lower elevation. So in these maps, red is higher complexity, blue is lower complexity. Another one that I used is curvature. So I did total curvature because curvature can be looked at in many different ways, and this is just kind of looking at the profile of the island and then also like aerial view. And then also standard deviation of the curvature. Ergosity kind of looks at how peaked your landscape is. So this is kind of looking at the surface area dividing by the planar area underneath. So on the left, that little example has a higher ergosity than the one on the right. And then a new one that many people are starting to use is the Compound Terrain Complexity Index, or CTCI. And this is looking at many different aspects of complexity. So it uses range and standard deviation of elevation, total curvature, and ergosity. So I mentioned that I'm going to mainly show the results for the snails because we have a higher spatial resolution for the snails since it's in our lab. And I took an additional step as well. So we have the age and isolation, an area of Galapagos, we have species richness for these snails, and then the complexity indices. But the additional step I took was I took satellite imagery of the Galapagos and tried to eliminate as much of the landscape that we know the snails can't inhabit. And so I did this by creating a normalized difference vegetation index, or NDVI. And the reason why I did this is because many parts of the younger islands in the Galapagos are lava flows. So we know that these are huge barriers for the snails, and we pretty much look at them as the ocean for the snails, because they're not going to be able to inhabit there. So to create NDVI, you take different bands from the satellite, and it gives you a kind of a measure of what has high photosynthesis and what has low photosynthesis. So by overlaying all of the points that we have of where snails have been collected over the last 16 years, we can then eliminate as much of the landscape as possible. So you can see for the younger islands, much of the landscape is gone, so we can see kind of what is important to the snails rather than looking at the entire landscape. So when I present the results, I'm going to show you the taxonomic group on the left, and then all the significant models that were run for the snail data sets, with HD standing for habitat diversity, and we use the number of native plant species for these islands as a proxy for habitat diversity. And then also all of the abbreviations for the complexity indices are Yannik. Then I'm going to show how they were significant. So, these were all significant, but red line is going to indicate the one with the lowest ASC. Blue lines will be delta ASC less than 2, and black lines will be delta ASC greater than 2. So we see when we look at the entire landscape for the snails, that while these models that include a topographic complexity index, they're significant, they weren't close. They had a very high ASC compared to the just habitat diversity. But if we look at just the landscape, we can see that the complexity index becomes much more important. So this needs to be considered when we start applying this to other taxonomic groups. So to kind of wrap it all up, I didn't show the results for if they follow a hump-shaped curve or not, but we did not see any of the complexity indices following this hump-shaped prediction. And while the verbal description makes sense, a lot of times people don't actually describe what they mean by topographic complexity. So in the geomorphology journals, there are quite a few different ways that people define it. It makes a pretty big difference. So you can have an island that has high elevation, which would be considered complex, but have very little curvature, which would be not that complex. So by adding a topographic complexity to the general dynamic model, we saw for 4 of the 11 taxonomic groups run that it was significant. And again, more work needs to be done because this was all done at one scale. So this, each of these taxonomic groups interact with the landscape a different way. So snails experience the landscape way different than, let's say, reptiles, because their range is going to be way different. But interestingly, if we use habitat diversity as the sole way of explaining species richness across the islands, we see that for 7 of the 9 animal groups that it is significant. So this maybe needs to be considered for future work, but a lot of places don't have a complete list of native species for their islands. And then also with the NDVI, I believe more work can be done with satellite imagery, since satellite imagery is being updated all the time, but we need higher spatial resolution for other taxonomic groups. So I want to thank my advisor, Dr. Christine Parent, postdoc in our lab, Dr. Andrew Kramer, and funding sources, but especially the Charles Darwin Foundation for downloading all of their data. Thank you.
So when you were calculating isolation, it looked like that was just the mean distance. Have you played with weighting that by island size?
I have not. I do want to look at other ways of looking at isolation, because if you have a bunch of small islands around you, it can affect what's going on. So yeah, area would be important, and also winds and other stuff like that. Any questions?